Okay.
Okay, we're ready?
Yep, we're ready.
All righty. Well, good evening, everybody, and thank you so much for being here. I want to welcome you all to the 2020 virtual 3-minute thesis competition. This is a new technology for us, and we're really excited to be sharing it with you this evening. And most importantly, we're really excited to be sharing the work of these amazing graduate students with you. UNC-Chapel Hill is such a collaborative and supportive university, and I think your presence here today is an example of the wonderful support that you provide for our graduate students. And I'm honored here today because this is really a celebration of graduate student achievement. Each one of our contestants has produced outstanding research and has done what must have seemed unthinkable to them a month ago. They've taken the knowledge that they've created in their graduate education and they've found a way to describe it in only 3 minutes using only one slide and to describe it in a way that a lay audience will understand. As you probably know, clear and concise communication in the professional world has never been more important than it is today. Each of our contestants has come through a very competitive process. There was a preliminary round. And so I want to congratulate our 10 finalists for making it this far. And we're really excited to hear your work today. And we should have a slide here to show you an overall view of the students who will be presenting today. Do we have that slide? Well, while we're pulling that slide, I'll tell you a little bit about 3-minute thesis. It's sometimes called 3MT, and it's an academic competition that, again, provides students with the opportunity to foster effective presentation and communication skills. 3-minute thesis was developed by the University of Queensland back in 2008. And in the intervening years, it's come to be held at more than 900 universities around the country. And those universities are in not more than 900 universities around the world. And those universities are in 85 plus countries around the world. So we're really pleased here in the graduate school to be able to sponsor a 3-minute thesis competition on our campus. And I want to give a huge thank you to Brian Rabarczyk, the Graduate School's Assistant Dean for Academic and Professional Development, for all the hard work he's put into putting this competition together. And we can all give him a virtual round of applause. Also, an enormous thanks to Rachel Underhill, who's helping us to manage all the complex technology that we're going to be using tonight to bring this wonderful event to you. So again, applause for Rachel. The winner of our competition will receive $1,000 and the runner-up will receive $600. And there's also going to be a People's Choice Awardee that will be determined by everybody in the audience, and that person will get a $400 award. In addition to getting the $1,000 award, the winner of our competition will also advance to a regional competition to represent our university against other 3-minute thesis winners from universities around our region. So in order for us to choose the folks who get these awards, of course, we have to have a wonderful panel of judges. And I'd like to acknowledge and thank them very much right now. I don't know if you'll be able to see them, but perhaps they can, maybe if they can hit their reaction button so you know who they are, or just kind of wave, then I can introduce them to you. Drusy French is an alumnus of the university and a cultural mythologist and an enormous supporter of graduate students whom we're very grateful to have here. Michael Lee is also an alumnus of the university, and he's a principal in Cardiff Consulting Services. Thank you so much for being here this evening, Michael. Kate Murphy is the higher education reporter at the News and Observer, and we thank you so much, Kate, for the time that you're taking to be with us this evening. Ann Rowe is a retired deputy provost from Florida State University. I don't think Ann has joined us yet, but we'll keep an eye out for bringing her in. Oh, there she is. I'm sorry, Ann, I missed you. Apologies. Jeff Warren is the executive director of the North Carolina Policy Collaboratory. Jeff, thank you for being here this evening. And Elaine Westbrooks is the vice provost for university libraries and university librarian. Thank you so much for your time this evening, Elaine. So the way this is going to work this evening is each student will give their 3-minute presentation, and then we'll give the judges a few minutes to make some notes and to think a little bit about their scores. Once all the contestants have presented their research, then we're going to take a very short intermission. And during that time, the judges will finalize their decisions. And after that intermission, the winners will be announced. But we have a really important job for you, those of you who are in the audience. You're the ones who are going to determine the People's Choice winner. And so as you listen to the presentations, Please take note of the person who's a crowd pleaser, the person who's telling the story that's most compelling and in the most compelling way, the person who connects most effectively with the audience. At the end of the presentations, we'll post a link for you to submit your, for you to cast your vote for the person who will be the People's Choice winner. And we're very, very excited about engaging you in this process in that way. So before we get into the presentations, just a few words about the rules of the competition. Again, each individual has 3 minutes for their presentation. They can only have one static slide, no transitions, no animations, no movement of any kind, and the slide is up from the beginning to the end of their presentation. There's no additional electronic media, no sound, no video, no props, no costumes, no musical instruments, no laboratory animals. Again, the presentations are limited to 3 minutes, and if anyone goes over the 3 minutes, then we have to disqualify them. Presentations are spoken word only, so please don't anybody break into song. The presentations are considered to have started when the presenter starts their presentation by either moving or speaking. The decision of the judging panel is final, and each participant will be judged on the comprehension and content, as well as the way they engage and communicate with the audience. So those are the ground rules for our very, very exciting evening. And again, we're so grateful to have you all online, so grateful to have our fantastic judges, and so grateful to these wonderful students who are going to provide us with a window into the exciting research they're doing here at UNC-Chapel Hill. So with no further ado, I'd like to introduce the first speaker, the first presenter of this evening. And that is Chi-Young-Wei Huang from chemistry. And the title of the presentation is Feeling is the Truth, Inhomogeneity in Materials.
Good. What do you see in this color image? I took this photo a few years ago when I visited Shanghai. The display of the thousands of candies is phenomenal. Yes, also sweet. But what really makes the eyeballs attractive is not only about the numbers, but how they pack to each other. You see, they have different sizes, shapes, some of them align in a vertical way, some of them in a horizontal way. All these difference, we call it inhomogeneous properties. In real life, all materials are inhomogeneous. The materials I studied can be used in our everyday electronic gadgets, from cell phone, laptop, to the batteries and solar cell that power our electronics. To study the inhomogeneity in the materials, I need a special tool to look at it. Otherwise, I will see information that's averaged out like the other half of the image. They are distorted, smeared out, and not real. To capture the real inhomogeneity, I use scanning probe microscope. There is a saying, seeing is believing, a feeling is the truth. And that is how scanning probe microscope works. Imagine you slide your finger across the surface of an object. You can feel the texture, shapes, all the information is transferred from your fingertip to your brain. And the concept is analogous to scanning probe microscope. Instead of a fingertip, I use a very sharp tip and bring in closely to the surface of the materials. All the information can be carried through the tip to the computer and get analyzed. The microscope is powerful when the materials at tiny length scales at nanometers. That is 1,000 times thinner than your single strand of hair. Why do we make it so small? Turns out they have a higher and better electronic performance. For example, the graphite in a pencil, we don't normally consider conductive. But when we peel, because electron doesn't want to move, but when we peel it off to a few nanometer thick, it becomes highly conductive. If we can use that material to the cell phone and laptop, it can be running at higher performance. If we use that to the batteries and solar cell, it can be running at a lower power consumption, and so we can save a lot of energies. The level of inhomogeneity can influence how they function in the applications. Just like the inhomogeneity in these candies, the size, shapes, packing structure all matters. To find out the real inhomogeneity, remember, feeling is the truth. Thank you.
Thank you, Chung-Wei. And now we'll give the judges just a few minutes to make their scores. But while they're doing that, I want to give you something to think about. And that's, I want you all to think about what might have happened in the last 3 minutes. So why don't we start by thinking about how many babies were born worldwide in the last 3 minutes? I'll give you a minute to think about that, and then I'll give you the answer. So I don't know if anyone was able to guess the answer, but it's 802 babies born worldwide in the last 3 minutes. Okay, so let's move on to our next presenter. And our next exciting presentation is by Miriam Oke. She's from Biomedical Engineering, and her talk is entitled Human Intestine on a Dish. Miriam, the floor is yours.
2 years ago, while I was assisting a pediatric surgeon in an operating room at UNC, I was overcome with emotion when we had no choice but to remove the entire large intestine of the 7-month-old that we were operating on. This child, not even 1 year old, had already gone through multiple surgeries and intestinal biopsies in an attempt to treat the symptoms of his complex gastrointestinal disease. It made me wonder, why don't we have better tools to study human intestinal diseases in vitro or outside the body? In my current lab, we are trying to do just that. Our lab has helped develop a protocol that allows us to grow human intestinal stem cells on a dish and keep them alive for months. We can do this by taking a small sample of intestinal tissue taken from a routine biopsy and then carefully processing that tissue to isolate stem cells from the sample. The amazing thing about these stem cells is that with the right environmental cues, they have the ability to become any type of cell in the intestine. We can then take these stem cells and reprogram them using genetic engineering techniques to make them easier to track in real time. Imagine that stem cells are like fireflies and that other kinds of cells are like other kinds of bugs. Until the fireflies light up, we can't really tell them apart from other bugs in the night sky. However, in our lab, we can reprogram the stem cells so that they glow a bright green color under the microscope, kind of like a firefly, which then enables us to distinguish them from other cell types on our human intestine on a dish. We have already demonstrated this successfully in our lab, and the next step is for us to further modify the cells so that they can change into different colors depending on what cell type they turn into. This way, we can culture and grow the cells on a platform with different compartments representing the human intestinal tract and the underlying bloodstream in the body. For example, in the upper compartment, which represents the digestive tract, we can add pill-based medicines and chemicals produced by gut bacteria, while in the lower compartment, we can add immune components and therapeutics like IV drugs. With these technologies, this enables us to simulate the human intestine outside the body, and like a firefly, will hopefully shed some light on the potential of the future of personalized medicine for gastrointestinal diseases. Thank you.
Thank you, Miriam, for that presentation. We'll give the judges a few minutes to do their magic, but in the meantime, I want you guys to be thinking about McDonald's and the number of things McDonald's might have sold in the last 3 minutes. How many hamburgers do you think they've sold? How many Big Macs? How many fries? And I'll give you that answer in just a couple of minutes. So if you're wondering, in the last 3 minutes, McDonald's sold 24,720 hamburgers, about 5,700 Big Macs, and over 34,000 pounds of fries. Now, these numbers are all pre-COVID, and quite frankly, I don't know whether they are increased or decreased after the COVID pandemic, but that gives you a snapshot of how much McDonald's sells in 3 minutes. Okay, let's move on to our third presenter. Our next presenter is Jelaya Clark from Environmental Sciences and Engineering. And the title of her talk is Eating for Two, Mother's Diet as an Intervention for Arsenic-Induced Lower Birth Weight. Jelaya, the floor is yours.
While much about childbirth seems magical, it's actually a tightly regulated process. During the first weeks of pregnancy, finger-like projections emerge from a ball of cells that will go on to form the embryo. These finger-like projections invade the wall of the uterus to form the placenta, connecting the baby's umbilical cord to its mother's blood supply. I know what you might be thinking. That sounds like a parasite, but proper placental invasion is critical to fetal growth because it allows for the transfer of nutrients. Despite these features, the placenta is somewhat of a double-edged sword, also transferring toxic chemicals circulating in the mother's blood. My research aims to protect developing babies from a particularly harmful chemical, arsenic. Arsenic is a cancer-causing chemical typically found in contaminated drinking water. It's known as the king of poisons and the poison of kings because it's been implicated in several murders throughout the Middle Ages. More recently, maternal arsenic exposure has been associated with lower birth weight in several research studies. This is concerning because low birth weight is associated with infant mortality and an increased risk of heart disease later in life. As seen in my slide, arsenic hinders placental invasion of the uterine wall, constricting the flow of nutrients and oxygen. Fortunately, arsenic metabolism relies on B vitamins such as folate that we typically consume in our diets. Among adults, B vitamin supplementation has been shown to improve the body's ability to remove arsenic and protect against arsenic-induced diseases. My research will evaluate whether B vitamins also protect developing babies from the harms of arsenic exposure. To test this, I'll use a unique data set collected from women who were exposed to arsenic during pregnancy. Using statistical modeling approaches, I'll evaluate whether the negative association between maternal urinary levels of arsenic and infant birth weight is attenuated or completely diminished by higher B vitamin levels. So far, I've seen that babies born to arsenic-exposed women who are also deficient in vitamin B12 weigh up to 50 grams less than their counterparts, who are also born to arsenic-exposed women with optimal levels of this vitamin. I'm so excited to complete this work as part of my dissertation because it has global implications. Around the world, hundreds of millions are exposed to arsenic through contaminated drinking water. This includes people living right here in North Carolina, where arsenic has been measured in drinking water at levels exceeding government regulations. My research aims to protect one of the most vulnerable groups and could inform the dietary recommendations made to pregnant women. In fact, it might change the meaning of eating for two. Thank you.
Thank you, Jelaya. So we'll give the judges a few minutes to work their magic. But while they're doing that, let's stick on the subject of food and drink. How many coffees do you think Starbucks has sold in the last 3 minutes? Do you give up? Well, in the last 3 minutes, Starbucks has sold nearly 17,000 cups of coffee worldwide. I kind of wish I'd bought stock in Starbucks a while back. Okay, our fourth presenter is John Myers, who comes to us from chemistry, and the title of his presentation is A Grain of Salt. John, the floor is yours.
You're outside eating the best burger and fries. It's hot and it's humid, but the food is worth it. As you sit back and relax, you notice that some salt on your plate looks a little wet. Is it melting? Actually, water is being pulled right out of the humid air, dissolving that salt crystal layer by layer until all that's left is a little droplet of ocean on your plate. This chemical process of liquefaction is called deliquescence, and the reverse process of crystallization is called efflorescence, and both are pretty savory topics in food chemistry. Now, it's not all about food. I work with a salt crystal that has applications in solar energy harvesting, medical X-ray imaging, and optical computing. But recently, researchers noticed something interesting. These crystals, called methylammonium lead iodide, are very dark, but in the presence of methylamine gas, they become completely transparent. And when removed, they revert back to their darker color. What is going on here? Why and how does it happen? Well, bring back the burger and fries. This looks suspiciously like deliquescence and efflorescence. To verify this, I built a computer-controlled methylamine reactor that controls temperature and pressure. And because the crystallization is particularly interesting, I built a custom microscope around the reactor that allows me to see when and where each crystal is born and how it grows over time. Each crystal grows as a perfect little square and grows and grows and grows until collision. After analyzing thousands of images and other data, I now know the exact conditions where liquefaction and crystallization will occur for a range of temperatures. And more importantly, I've calculated the fundamental driving forces for the reaction. This is, in fact, a new type of deliquescence and efflorescence. Because of this fundamental understanding of the what, the how, and the why, I can now liquefy small crystals and make new ones more than 10,000 times their original size. These large crystals efficiently absorb light, and when given energy, they can emit very bright light. So with one of these crystals, I created a highly responsive light detector. And next, I plan to use them as LEDs, lasers, and more. Certainly, the best technologies that we use today exist because someone began asking the right fundamental questions. And when answering those questions, I've learned that the most simple explanation is usually the right one. All the others I take with a grain of salt.
Thank you, John. I don't envy the judges. They're going to have a tough decision to make. But in the intervening time while they're making that decision, why don't we think about Facebook? How many photos do you think have been uploaded to Facebook in the last 3 minutes worldwide? The answer in a moment. And the answer is just shy of 729,000. That's a lot of photos on Facebook. And I'm thinking that's one of those statistics that's probably increased during the pandemic. Okay, our fifth presenter is Isabel Laterzo from Political Science. And the title of her talk is Don't Call the Police, Measuring the Dark Figure of Crime. Isabel, the floor is yours.
Imagine you're robbed. You're probably upset, confused, but you likely also want something, help. The source of that help for many people is obvious, the police. But for many others, depending on your individual relationship with the police, your community's history with them, and a variety of other factors, the answer to that question might not be so obvious. This is particularly politically relevant today. I decided to begin tackling this problem and understanding how it translates into often inaccurate and unreliable data. Because of this underreporting, we often have statistics that do not reflect reality. This is called the dark figure, or the difference between levels of crime recorded by the police and true levels, which are difficult to measure. Many practitioners and researchers and NGO officials tackle this problem constantly when they're trying to make assessments about crime and violence, but know that their data is often questionable. I decided to begin examining this problem in the context of Latin America, where we know that relationships between citizens and police are tense. In particular, I look at Chile and a nationally representative survey, which asks each year people's individual victimization history. Using this survey and a newly developed method from 2019 called BARP, I estimate the true victimization rate for 101 different communities across Chile. This represents the entire urban population, or about 60% of the country's total population. I then compare these true rates to official statistics recorded by the government, and I find that underreporting is massive. As can be seen on my slide, in La Pintana, a small community in Santiago, Chile, we see that official statistics tell us that there's only about 8 victimizations for every 1,000 residents. But according to my predictions, it's actually closer to 200. This is a 96% underreporting rate, or a dark figure of 96%. Now, why is what I've done unique? Well, it's unique because typically the nature of the data does not allow us to make such fine-grained predictions. We can only do it at the regional or national level. But with this new method, we can understand which communities have patterns that explain underreporting and which do not. This opens up massive opportunities for government officials and researchers like myself. And I began to apply this method to understand how we can explain this variation. I gathered more data from the government of Chile regarding the amount of police officers in each community and found that, interestingly enough, the number of police officers and the amount of reporting that goes on have no relationship. This is contrary to much general knowledge that more police equals more security and prevention. This might not be the case. Rather, I find that when there's more security cameras and clear evidence that victims were a victim of crime, they're more likely to report their situation to authorities and seek justice. Thank you very much for your time.
Thank you, Isabel. So we'll give the judges a few minutes again. And while they're doing that, if we talk about Facebook, we have to talk about Twitter, right? So can you guess how many tweets were sent in the last 3 minutes? And the answer is a little over 1 million. So slightly more than 1 million tweets were sent in the last 3 minutes. Okay, our sixth presenter is Odessa Gowdy. She's from Biochemistry and Biophysics. And the title of her talk is from computer models to cancer drugs, creating inhibitor proteins to combat cancer. Odessa, the floor is yours.
Imagine that you were asked to design a full-scale replica of the Eiffel Tower made exclusively from Legos. What would your first reaction be? Would you be excited about all the different design possibilities? Would you be worried about the structural integrity? Would you consider the shape of the arch? Surprisingly, these are all similar questions to Instead, I'm engineering safer cancer therapeutics from protein building blocks. And to sift through the hundreds of thousands of different design possibilities, I'm using a computer program that models the best designs as measured by shape, structural integrity, and interactions to other proteins. I'm designing a safer cancer therapeutic because the current drugs do not discriminate between a target marker on a cancer cell versus an identical marker on a healthy cell. This marker sends a message to the immune system that the cell is normal, which is false when it's coming from a cancer cell. And recognizing the difference between a true normal cell from a false one is important to preventing the many toxic side effects patients suffer due to their cancer treatments alone. To overcome this problem of toxicity, my drug starts in an inactive state and only switches to its active state once it's found a cancer cell. And this switching activity is due to the presence of an inhibitor. This inhibitor, shown here in yellow, is computationally designed, and it sits at and deactivates the binding site of the protein that will eventually bind the cancer cell marker. And these two components are linked together to form the overall drug. Now, as long as that inhibitor is present, the drug is forced into its inactive state while traveling through the body. Once the drug reaches the cancer site, the linker is cut in half and the inhibitor falls away, leaving an active state drug which can then bind nearby target markers. And once bound and hidden, those target markers can't send false messages to the immune system, meaning that the immune system can successfully come in and destroy the cancer cells. In my research, I've shown that the presence of an inhibitor successfully forces the drug into its inactive state. Moving forward, I would like to show that the loss of the inhibitor switches the drug into its active state only once it's found a cancer site. And finding an inhibitor that can play both roles is going to be pretty tricky, but that's why we're using this computer program to predict the best designs for us. What truly excites me about my research, and what I hope I've convinced you of today, is that you can start with a tool as simple and as accessible as a computer or a laptop to design safer cancer therapeutics with the ultimate goal of maximizing patient safety. Thank you.
Thank you, Odessa. So we'll give the judges time again. And if we've talked about Twitter and we've talked about Facebook, we got to talk about WhatsApp too. So how many WhatsApp messages do you think were sent in the last 3 minutes? The answer in a moment. Does anybody have a guess? Well, if you guessed about 87 million, then you're right. About 87 million WhatsApp messages were sent in the last 3 minutes alone. That's a lot of messages. Okay, our seventh presenter is Jonathan Slosser from the Hussman School of Journalism and Media. And Jonathan's title is Disinfectant, News, and Twitter. Jonathan, the floor is yours.
Can you hear me? Can you see me? Can you see me? Okay. On Thursday afternoon of April the 23rd, during a White House press briefing, President Donald Trump presented a misinformative statement suggesting the use of disinfectant as some sort of injection to battle the coronavirus. The quote can be seen in my slide. This statement, while likely unintentional or just the outcome of a moment of riffing, is a prime example of misinformation and not only poses traditional risks, but also poses a public health risk, as we don't really want anyone ingesting disinfectant. Since misinformation can directly influence individuals' attitudes and behaviors, understanding how this misinformative statement is received and discussed by the public becomes a serious priority. Well, after Trump made this statement, Twitter and the news media exploded and offered the perfect opportunity to do some research. To begin, I first collected, cleaned, and processed thousands of tweets and news media articles. Once I had that initial data processed, the challenge became identifying a way to systematically conduct the analyses. Considering that language is extremely variable and meaning can come about in multitudinous ways, systematically examining language requires some special considerations and procedures. With that in mind, I employed topic modeling approaches, specifically a structured topic model, to classify the data into topics, to identify the texts and terms that contributed most to a topic, and to examine how topics changed over time. With this approach, I actually found some interesting things. First, I found that the terms used within the two discussion realms differed, such as the more frequent use of the term bleach on Twitter compared to the more frequent use of the term disinfectant within the news media, thus revealing an initial mismatch as to how the topics were being discussed, and a mismatch in the specific language being used. Secondly, I found that there were differences in the actual topics being discussed. On Twitter, the major themes focused on correcting the misstatement, criticizing Trump for making the statement, and/or criticizing the media for taking the statement out of context. But within the news media, the conversation focused on how Trump was lying, presenting false information, or directly causing harm to the public. Third, I found that the evolution of conversations differed as well, with the conversation gradually descending on Twitter compared to an almost immediate descent in the news media, as if the conversation immediately halted once the news media moved on to the next story. Overall, this work engages with the mismatch between the conversations taking place on Twitter compared to the conversations taking place in the news media, and it reflects findings in similar work examining the differences in how the media reports issues and on how the public engages with those issues. With current concerns around misinformation in social media, this area of work is more necessary than any other time previously. But this type of research is just starting the beginning, and it will be fascinating to see what we can discover in the future. Thank you for your time.
Thank you, Jonathan. And so we'll let the judges get to work. And while they're working, during this pandemic, I think a lot more of us have been ordering a lot more on Amazon. So I'd like you to think about how many Amazon orders have been placed in the US in the last 3 minutes. The number is 833 orders. That's one where I wonder what the differential is going to be before and after pandemic. And when things go back to normal, whatever that is, whether that number will normalize as well. Okay, our next presenter is Rachel Johnson. She's from chemistry, and the title of her presentation is a hybrid antibiotic to overcome antibiotic resistance, Rachel, the floor is yours.
Have you ever gone to the doctor and been prescribed an antibiotic? You likely have, and this is because you had a bacterial infection. Antibiotics are used to kill or inhibit the growth of bacteria. Now, up until 100 years ago, if you had a bacterial infection, you likely wouldn't survive because the first antibiotic wasn't discovered until the late 1920s. But today we're faced with a different challenge. Numerous antibiotics have been discovered, yet bacteria have become resistant to these drugs. In fact, there's bacterial infections that cannot be treated by any known antibiotics today. This is a global health crisis. In the United States alone, over 35,000 deaths per year are from antibiotic-resistant infections. About 30% of these deaths are from methicillin-resistant Staphylococcus aureus, or MRSA, which can cause infections of the skin, lungs, and even the blood. Mepiricin is an FDA-approved drug that's used to treat topical MRSA infections. However, prevalence of mepiricin resistance has increased clinically. As you can see on my slide, the red bacteria are able to be killed by mepiricin. However, the blue, or mepiricin-resistant bacteria, survive, are able to replicate, and allow the infection to persist even after antibiotic treatment. But I don't want to give all bacteria such a bad reputation because they don't all cause diseases. There's bacteria in the soil that help plants to grow. There's bacteria in our guts that help us to digest food. And there's even bacteria that make antibiotics themselves. I work in the Bowley lab on a small molecule, thiomarinol, made from marine bacteria. As you can see on my slide, it has an interesting structure. It's actually two antibiotics linked together, as I've highlighted in red and blue. The red portion of the molecule is extremely similar to the FDA-approved drug mepiricin, but notably, thiomarinol is able to treat MRSA 100 times more effectively than the FDA-approved drug. And you can see that this is a hybrid antibiotic, and it's extremely rare to find in nature. My research shows that thiomarinol is able to inhibit an essential metabolic process in bacteria, protein synthesis. Now, like mepiricin, it does that by binding to an enzyme and halting its function. But thiomarinol binds to the enzyme 1,500 times better than the FDA-approved drug. Even more, thiomarinol is able to treat bacteria that are resistant to mepiricin. We have evidence to show that the blue portion of the molecule allows thiomarinol to overcome mepiricin resistance by acting through a different mode of action. Overall, we envision that our work on thiomarinol can be applied to the design of new hybrid antibiotics. And ideally, we could use previously discovered antibiotics and repurpose them to make new hybrids to fight antibiotic resistance. Thank you.
Thank you, Rachel. So we'll give the judges a few minutes, and why don't we think about the number of Uber trips that have been completed in the last 3 minutes? The answer is about 80. 80 Uber trips completed in the last 3 minutes. Again, a statistic that is likely going to be heavily influenced by the times in which we are living. Our next presenter is Danielle Weber from Psychology and Neurosciences. And the title of her presentation is Emotional Arousal in Couples Predicts Short and Long-Term Relationship Satisfaction. The floor is yours, Danielle.
When we think about predictors of health, we tend to focus on the individual, but one of the best predictors of health and well-being is the quality of one's relationships. When we're in a good relationship, it brings out the best in us. And a bad relationship is a major risk factor for mental and physical health difficulties, even mortality. So it is of utmost importance that we understand how to promote good quality relationships. And a big part of good relationships is good communication. When communication gets disrupted, that leads to escalating conflict and that degrades the relationship over time. So communication is central, but it's often been studied by looking at behaviors, and a lot less has been studied about emotions. Emotions are often thought of as individual experiences, but they're not. If you express your emotions to your partner, then your partner then reacts, and then their reaction can then change how you feel. So emotions change a lot within a person as well as between people within an interaction. And that's the focus of my research program, how we understand emotional exchanges and communication and how that relates to well-being. So I had the opportunity to work on a longitudinal data set of couples where they completed one interaction at the beginning of the study and then were followed up 25 years later to see how happy they were in the relationship. So within this interaction, I was interested in one indicator of emotional arousal communicated in the voice called fundamental frequency, which is essentially the pitch of your voice. So we hear pitch go up and we hear that as someone being more emotionally aroused. So what I did is I looked at some algorithms and advanced statistics to track how these values of pitch change across the conversation, and then look at how that predicts relationship satisfaction 25 years later. So what I found is that indeed, subtle changes in pitch in one interaction could predict relationship satisfaction 25 years later. Specifically, what I found is that if one person was increasing in arousal across the conversation, they were happier in the relationship, but their partners were less happy 25 years later. And also found that for women, if women increased in arousal and stayed there longer before coming back down, they were happier, but their male partners were less happy when women express those emotions. So what this is telling us is that we're in a difficult place in terms of how to intervene on couples communication, because it's not the case that one size fits all. Instead, we need to find a way to balance the emotional needs of both people. How do we help the person expressing themselves, particularly women, do so in a way that's genuine and true to them, helps them process emotions, and in a way that the other person can really understand? And we help the person receiving those emotions of how to increase their capacity for understanding and hearing those elevated emotions in a way that doesn't reflect negatively on them for the relationship. So this tells us that we need to have a nuanced approach for understanding communication for the betterment of the relationship overall. Thank you.
Thank you, Danielle. So we'll give the judges some time, and I'm going to give you a two-parter this time. The first part of the 3-minute trivia is, what's the busiest bridge in the world? And the second one, of course, is how many cars have driven over that bridge in the last 3 minutes? The answer is in a moment. I'll give you a hint on the bridge. It's on the east coast of America. So the answer is the George Washington Bridge, and 575 cars have driven over the George Washington Bridge in the last 3 minutes. Just a little bit of bridge trivia for you. So our last presenter is Ricky Pimentel from Biomedical Engineering. And the title of Ricky's talk is Muscle Metabolic Energy Costs While Modifying Propulsive Force Generation During Walking. Ricky, the floor is yours.
I'm Ricky Pimentel, and my goal is to help people age gracefully so they can stay active and healthy throughout their lifetime. Compared to the young, older adults require more energy to walk at the same speeds, reducing their endurance and independence. A major theory on this higher metabolic cost is the shift of muscle work from muscles spanning the ankle to those at the hip. When shifting muscle work, older adults tend to rely more on muscles with longer fibers and shorter tendons. This requires older adults to perform more active muscle contraction and less passive tendon stretch and recoil. This may lead to higher metabolic costs by literally taking the spring out of their step. When young adults push off the ground with less force while walking, they redistribute their muscle work just like older adults. So we wanted to test this shift in muscle demand theory in young adults because we can control for other age-related changes that affect walking. It's not really feasible to measure the metabolic cost of individual muscles during walking. So we used musculoskeletal models to calculate the muscle forces required to generate movement, and then estimate the metabolic cost of each muscle individually, accounting for muscle fiber length, activation level, and tendon elasticity. We asked young adults to walk at their typical speed when targeting their normal push-off force, as well as 20 and 40% lower than normal, shown in blue. They also completed trials of 20 and 40% larger than normal push-off force, shown in orange, but I won't discuss that data. We recorded their motions and forces during each of these trials and also measured how much oxygen they consumed to calculate their actual metabolic cost. Our model estimates explained about 1/3 of the variance in directly measured metabolic cost. The cost of walking increased when targeting smaller push-off forces, just like older adults, mainly explained by muscles spanning the hip and the knee. At the hip, the increased costs mostly occurred during early stance. With less ankle push-off, the leading limb's hamstring muscles have to work harder to pull the body forward and maintain forward progress with each step. During leg swing, targeting smaller push-off forces led to higher metabolic costs in muscles spanning the knee. Here, the quadricep muscles had to perform more work to swing your leg forward and straighten it before the next foot strike. Our work highlights the importance of ankle push-off during walking and how movement compensation to other joints leads to higher metabolic costs, even in young adults. We need to find ways to maintain ankle power capacity using rehab therapies or assistive devices. Ultimately, this would lower the metabolic cost of walking for older adults so they can keep moving forward with active and healthy lives.
Thank you, Ricky. And thank you to all our presenters. I hope you'll join me in giving them a huge virtual round of applause. And I'm sure you'll agree with me that these presentations were absolutely fantastic and that there's amazing research going on here at the University of North Carolina at Chapel Hill, and that graduate students are playing a major, major role in it. And what an amazing job these graduate students have done in describing their research to us in ways that those of us who aren't in their fields can understand. So we're going to take a 10-minute break while the judges do their very, very difficult job of choosing the first place winner and the runner-up. But you have a job to do too in the audience, and that is you need to help us choose the People's Choice winner. You'll see on the screen now a link that you can go to enter your choice for the People's Choice winner. So think about the presentations you've heard, think about all you've learned in the last 40 or 50 minutes, and please make your choice. And we'll see you again in about 10 minutes time. Thank you.
Okay, Suzanne, what do we do now?
I think you're supposed to be going into a
I was not sure about hitting leave. Yeah, give me the leave. I'm here.
Okay.
So the moment we've all been waiting for is just about upon us, but I'm gonna leave you with one last fact on 3 minutes. Over the last 3 minutes, about 196 million liters of water flowed over Victoria Falls, about 270 million liters over Iguazu Falls, and about 102 million liters over Niagara Falls. That's a whole lot of water in a very, very short period of time. And just imagine, there are times when it's so cold in upstate New York that Niagara Falls freezes. Before I announce the winners, I just want to give a few shout outs and thanks again. First of all, I want to congratulate all of the presenters today. I was in the room with the judges, and I think we all agreed that these were just amazing presentations, and that our students here at Chapel Hill are doing amazing, amazing work and have such wonderful presentation and communication skills. So huge kudos and congratulations to all of you. And thank you very much for participating in 3 Minute Thesis. I also want to thank all of you who've joined us online. This is the first time we've done a virtual 3 Minute Thesis, and I hope you'll agree that it was an exciting event. It was an important event. I hope you learned a little something this evening, and I hope you enjoyed hearing these wonderful presentations. And we'll get to your People's Choice Award in just a few minutes. Huge thanks and kudos to our judges. Thank you so much for taking the time out of your busy evening to be here with us and to help us make this very, very difficult and important choice. We're so grateful for all you do to support graduate students and graduate education at Carolina. And thank you for going the extra mile this evening with us. And finally, enormous thanks to Brian and to Rachel. Rachel for making sure that the technology works for us and keeping us on track, and Brian for organizing this event and for organizing the preliminary rounds that resulted in the wonderful presentations that you saw this evening. So with that, I think we can get a drum roll going. I don't know quite how to do that virtually. And the runner-up is Miriam Okay. Congratulations, Miriam. And now the winner is Jalaya Clark. Congratulations, Jalaya. Finally, the People's Choice Award winner is, congratulations, Isabel and Rachel. And congratulations to all the students who competed this year and all the finalists. Thank you again to all of you who participated this evening. I hope we'll give a round of applause to all of our winners, to all the presenters, and to you, the audience, for participating. Remember, 3-Minute Thesis is an annual event. Hopefully this time next year, we'll actually be all together in the same room. But whether we're in the same room or we're virtual, please remember that the students are here doing this wonderful work, and that we welcome the opportunity to celebrate their accomplishments with you. Again, congratulations to all the winners, congratulations to all who presented, and thank you all for participating this evening. I wish you all a good evening. Bye-bye now.